Hello and welcome. Today we are going to look at the sales cycle, and by the end of this tutorial you should be able to answer the following questions. One, what is the sales cycle? Two, what is the importance of the sales cycle? Thirdly, you should be able to listen and explain the various phases of the sales cycle. And lastly, you should be able to state how the sales cycle is controlled. So today's tutorial promises to be very much indeed interesting with a lot of information. Remember to please subscribe to our channel in case you've not yet done so. Let's get started. Before we go on to define the sales cycle, it will make more sense if we refresh ourselves about some basic information. Remember, the cell being the basic and functional unit of life is divided into two broad categories. That's the eukaryotic cells and the prokaryotic cells. The eukaryotic cells can further be broken down into subcategories. That's the animal cells, the plant cells, the fungi, and the protist. Animal cells can be divided into two subcategories, that's the somatic cells and the gametes or the sex cells. These two broad categories of cells reproduce through different means. For example, the eukaryotic cells will do so through mitosis, that's for somatic cells will divide through the process of mitosis, and the gamete or sex cells will divide through the process of meiosis. However, there are events that lead to this division, and the events or those events in combination with either mitosis or meiosis will constitute the entire cell cycle. For the prokaryotic cells, we do so usually through binary fusion, right? For this tutorial, our focus is not going to be on the prokaryotic cells, neither the plant cells, fungi, or protists. Rather, we are going to look at the animal cells, meaning those events that go on before we have the process of mitosis or meiosis. Let's move on. What is the cell cycle? The cell cycle can be likened to the development of we humans. What do I mean? Remember, at first, a kid or a child is born, right? That child goes on and reaches puberty, where he or she starts to develop some sex characteristics, like secretion of hormones that will make them very much indeed reproductively active. That child now develops into an adult, and when he or she develops into an adult, they go on to start to produce their own children, and their children will grow to or get bigger and start to produce their own. So you notice that the entire event is in the form of a cycle. That is just similar concept that applies to the cell cycle or to cells. The cells will be reproduced, they grow or develop, and then they reach at a stage where they themselves are capable of reproducing. So, in a simplified term, the cell cycle is defined as a regular sequence of events that produce new cells. So, as I have just stated, for the cell cycle, let's consider this, right? Remember, you have event 1 going on wherein the cell is produced. Even two, the cell starts to get mature, meaning it starts to produce things that will enable it to undergo division. At stage 3, the cell actually starts to prepare for division. Stage 4, the cells divides, right? When it divides, you have the daughter cell starting the cycle again at stage 1 and then going on and on and on and on and on. That's how the entire cycle is. So the events usually that constitute all of the activities going on here can be grouped into different phases of the work cell cycle, meaning events that occur in the stage 1, stage 2, stage 3, we can group them into a particular phase of the cell cycle. And just to state, the cell cycle is divided into two phases. Stage 1 here, stage 2, and stage 3 will constitute a particular phase. How will we call that phase? This is called the interphase, and it's basically the longest phase of the cell cycle. The next phase of the cell cycle is the M phase, also called the mitotic phase. Considering the cell cycle interphase, there are major subphases or stages that occur under this phase. What are they? First, we have the G1 phase, which is the stage 1, the G2, or the second stage, which is the synthetic phase, and the third phase, which is the G2 phase. When it comes to mitosis, we are not really going to delve deep into that for this tutorial. In subsequent tutorial, we will look at the stages of the mitotic phase, meaning in somatic cells, we have mitosis, and in gametes, we'll discuss meiosis. But for today, we are just going to focus on the interphase aspect of the cell cycle. So this phase is very, very much important, as I just stated. It actually distinguishes the gametes from the normal body cells that we have. So let's now move on and look at each of these here phases that occur in the interphase of the cell cycle. Let's now begin our study of the interphase stage of the cell cycle by looking at the very first stage. That's the G1, or the growth or the gap phase of the cell cycle. You hear me using that, right? There are so many names this phase is called. The gap 1 phase has certain primary information that you need to know about the cell cycle. What are they? The first thing is this phase is the longest and the most variable part of the cell cycle, meaning for different cells, this phase might be longer and it will have different length of time. The phase immediately occurs after mitosis. The moment you have the cell dividing, the next phase that comes out is the gap 1 phase. Next, this phase is primarily dedicated to certain important activity. Like I said, right, in our introduction, the cell primarily grows in this stage of the cell cycle. And also the nucleic acid and proteins that are needed for DNA replications are produced in this phase also. And lastly, the metabolic activities of the cells are carried out in this phase. What do I mean? For example, you have cells that are usually adapted for certain functions in the body. Like maybe you have the kidney cells, right? Or the cells that form the kidney, the renal cells. You have the cells that form the bladder. You have the cells that form our nervous system, the neurons. At this particular stage of the cell cycle, those cells start to carry out their functions that they are meant to carry out. And lastly, the cell cycle or this stage lasts for about 8 to 10 hours. So it's very much indeed lengthy. With that now, there is a point that restricts the entry of cells from the G1 phase to the S phase. This is usually called the restriction point. So if a cell fails to reach this restriction point, it means that that cell will not enter the S phase, right? But however, there is also another thing. If the cell fails to reach this restriction point, meaning all of these activities do not go on, like cell growth, the nucleic acid synthesis, the cell will enter into what we call a G0 phase or a gap outside phase. What is this phase all about? At this phase, the cell is usually dormant, right? An example of cells that are formed in this phase, you have the nerve cells, the muscle cells, and the liver cells. Remember, the nerve cells usually they reproduce while the fetus or the child is developing. But at that stage, you don't have any new neurons being formed. That's just the reality. Similar concept applies to muscles and liver. The liver cells they undergo low reproduction rate, and most of them are in this gap nerve phase. But what happens when there is an injury to the liver, or maybe we have a transplant? These guys are stimulated out of the gap phase and they replicate at a faster rate. The next thing is this phase might be temporary or permanent. Temporary in the sense of liver cells, for example, like what I said, right? Wherein we have an injury, these cells will leave from that phase and start to multiply again. For neurons or muscle cells, when they enter the gap nerve phase or the gap alpha phase, they remain there permanently. So for cells that stay there temporarily, how do they leave the gap or the G naught phase? Let's look at the various steps that go on here. The first thing is there are protein signals called mitogens or growth factors that are found in the extracellular environment. So these factors, when produced, they will bind to the receptor of the cell that is in the G naught phase. Upon binding to the receptor of the cell, they stimulate the kinase signaling cascade, and this in turn will trigger the cell to immediately start carrying out the necessary processes that we saw here that will enable it to reach the registration point, thereby entering the S phase of the cell cycle. So those are basic information you have to remember of the gap 1 phase of the cell cycle. Let's move on. Next, we're going to look at the last two phases for this particular tutorial. That's the X phase and the G2 or the gap 2 phase. When we're talking about the S phase, there are major events that occur here. And I'm going to just give you a brief description of this particular phase as you can see. This phase immediately occurs after the G1 phase. So after the cell has grown up and also all of the needed materials that are meant for cell division have been produced, this cell then enters the S phase of the cell cycle. Next, the phase lasts for about 8 to 12 hours in most cells, right? But one thing you need to note is the length of time it takes this phase actually depends on the genome of the cell. So if the genome of the cell is very much indeed large, you notice that it's going to take a longer time for the DNA and other machineries to be replicated as opposed to the genome being smaller, it's going to take a lesser time to have the various DNA and machineries to be replicated. So, based on the length or the size of the genome, you will have a variation in the length of time taken by the S phase. Also, leaving the S phase, there is a registration point, meaning the DNA has to fully replicate. All of the histones need to be present before we can have that particular cell leaving the S phase. If those things are not in tight, the cell will be kept in the S phase, and another factor which we'll discuss later will be triggered. Now let's look at a G2 phase, right? This occurs after the S phase, and it actually involves the formation of the centrioles, the proteins that are necessary for cell division, and it lasts about 2 to 5 hours, very short. Those are the basic information or basic things that occur in the G2 phase. Leaving a G2 phase to the mitotic phase, there is also a registration point wherein everything needs to be intact before the cell can divide. So if you notice from the onset, there are so many things that regulate the cell in dividing. Meaning if we don't want too many cells There are places that we can regulate the cell to prevent the cell from overproducing, or if the cell is not really up to task, there are ways to that we can regulate that. When it comes to the mitotic division, remember there are really subphases involved here. Like I said, we are going to look at those phases in detail in our next tutorial. Next, now let's look at how this particular cell cycle can be controlled. Talking about the control of the cell cycle, there are several factors that control the cell cycle. At first, we've already talked about this zero restriction point, right? Remember restriction point 1 between the G1 phase and the S phase, restriction point 2 between the S phase and the G2 phase, point 3 and so forth. Aside from that, there are certain family of proteins called the cyclins that actually function in the control of the cell cycle. So the cyclin, in addition to the cyclin dependent kinase, all function together to regulate the cell cycle. How does it happen? Let's look at the mechanism. Remember, or just know that there are different types of cyclin, meaning the family, right? You have type A, type B, type C, and type D. Whereas the cyclin dependent kinase also has different types: type 1, type 2, type 3, type 4, type 5, up to type 6. Each cyclin here activates different cyclin dependent kinase. And when that happens, the activated cyclin dependent kinase will phosphorylate specific proteins in the cell. When those proteins are activated, they will trigger the next phase of the cell cycle. Let's take, for example, right? You have the cyclin in the G1 phase. They bind to the cyclin dependent kinase in the G1 phase and trigger certain enzymes that are responsible to synthesize or carry out every activity here. By triggering the enzyme, it enhances the cell to now enter the S phase of the cell cycle. So after carrying out their activities, the cyclins are then removed from the cell by the proteasomes. Let's now go on and actually look at these steps in detail. Alright, let's begin with the G1 phase. So at the early stage of the G1 phase, you have cyclin D binding to either cyclin dependent kinase 4 or 6. When this happened, the G1 activities are stimulated, and meaning you have the cell really carry out metabolic function, you have the cells carrying out all set of activities. So at the early G1 phase, you have those processes being stimulated, right? Also, in addition to that, you have the cyclin A being synthesized. So by binding or the binding of cyclin D to cyclin 6, we actually synthesize cyclin A. In the late G1 phase, cyclin E binds to cyclin dependent kinase 2, and this actually triggers the entry of the cell into the S phase. Now let's look at the S phase. The S phase has what? Cyclin A. When you notice here, cyclin A was synthesized where? In the G1 phase. And now in the S phase is being used. So cyclin A binds to cyclin dependent kinase 2, and this will allow the cell to actually leave the synthesis phase and enters the G2 phase. Also, by binding of this to this will trigger the production of cyclin B. Let's go on. And lastly, in the G2 phase, cyclin B binds to cyclin dependent kinase 1 and allows the cell to leave the G2 phase and enter the mitotic phase. That's just the basic information about that. For the mitotic phase, before the cell can divide or before the cell can be allowed to enter the G1 phase, meaning you should have the spindle apparatus present. If they are faulty, the cell will not leave the mitotic phase because they are very much indeed important for cell division. Also, for chromosomes that are sticking together, the cell will not be allowed to enter or leave the mitotic phase. And lastly, in this phase also you have, a lot of regulations going on, right? That's just it for the mitotic phase. Like I keep saying, we are going to look at these phases in detail. Next, as in addition to the restriction we just listed, the cell cycle can also be controlled through or be affected by other external factors like the insufficient nutrients for cells to really go on and reproduce. If there are no nutrients, the cell cycle will be restricted. If the environment is inappropriate, you also have the cell cycle being affected. And lastly, if the DNA is damaged, the cell cycle will be prevented from proceeding or progressing to the next stage. Next, we talk about various restriction points, right? If you can recall, restriction point 1, 2, 3, 4, and so forth. If a cell is kept at any of those restriction points, and the reason for which it is being kept there is not solved. Let's say, for example, the cell has a DNA damage and it cannot progress from G1 to S phase, or it cannot progress from S phase to G2 phase. The cell or that particular damage should be repaired. If it is not repaired, proteins that are encoded by the tumor suppressor gene is activated. When those proteins are activated, they actually redirect the cell activity towards cell suicide known as apoptosis. So I was telling you, right? Let's take, for example, in the S phase, if there is a damage to the DNA and the cell cannot leave the S phase, it stays there for long, there will be a trigger of the process of apoptosis known as what? Self-suicide. There are many genes actually that code for proteins that are involved in the cell cycle control. These genes are generally referred to as the proto-oncogenes. So when there is mutation in the structure or expression of this gene, they get converted to the oncogenes. And those oncogenes actually, they do not have the capacity to control the growth of the cell. As a result, the cells go on to divide uncontrollably and might lead to cancerous cell. So the main reason, one of the major reasons we study the cell cycle is to know how to treat cancer cell, know how cancer cells actually replicate, and also how they can be restricted, how their growth can be restricted. If you take a look at most cancer drugs, they will actually act by restricting one of these points in the cell cycle. Like you take, for example, a methotrexate, you usually see it in the S phase of the cell cycle. So thank you very much for watching today's tutorial. I hope the information provided was very much indeed helpful to you. Check out our next tutorial, which will be talking about the process of mitosis and meiosis. And don't forget, if you've not yet subscribed, please subscribe to our channel and give us a like. Thank you very much. See you in the next tutorial.